very last presentation for today. This is by Trisha uh, Panganiban on the dietary carbohydrates, increase heat stress resistance of honeybees foragers.
Afternoon. And before I start, I do just want to say how great it was to be surrounded by so much knowledge today and to hear about all the new advancements in the bee community. Without further ado, let me introduce myself. So my name is Trisha Panganiban. I'm a fifth-year biology major, women's studies minor at the California State University, Los Angeles. This summer, I was able to participate in a research study within the National Science Foundation located on the island of Lesbos, Greece. And so before I get into the research, I do want to set the stage for where this research takes place. Like I mentioned, it was on the island of Lesbos, which is an island in Greece, and it is known for the large biodiversity that it has on this island. And interestingly enough, Aristotle actually stopped by this island while he was amidst his philosophing, and he studied the biodiversity and the different characteristics of each organism on this island. So with that being said, there is a rich scientific research history on this island. As you can see, there's a large biodiversity from these photos. And now that I've gone over where this research takes place, I want to go over the background and actually get into the study now. So we did focus on the nutritional aspect of how bees can tolerate heat stress. I forgot to mention this on the first slide, but usually with this internship, we were looking at different stressors and how this can better affect their heat tolerance. And so my study focused on the nutritional aspect. And with the first portion of this study, we looked at fed versus unfed bees, and they were fed with a 50% mixture of sugar water. Next, we looked at fed sugar water versus water-fed bees, and this was largely to control for any particular effects if there was any of dehydration. Next, we looked at rich versus poor nutrition, and this was done through looking at their consumption of bee candy. This was done with 2 commercially available bee candies available on the island at the time. And with the rich nutrition, we looked at a bee candy that was pollen and sugar, while the poor nutrition was a source of just sugar, so sugar paste. And then last but not least, we looked at high versus low sugar concentration and how this might affect their heat tolerance. So our hypotheses were, although we had multiple parts to the study, our general hypothesis was that with the access to carbohydrates, the honeybee foragers would be able to better tolerate any heat stress. And this might be in part because of their need to thermoregulate, their need for the high energy expenditure associated with this. So we were expecting that with carbohydrates as a source of energy, they would be able to better tolerate any heat stress applied. And so going into our methods, we first collected the bees. We had two hives, and they were trained to their associated feeders with particular scents. So we were able to control the source of the bees, and at least with having those 2 hives, we were able to compare and contrast. And if in case there was an issue with one hive in particular, then we were able to look at the other hive and have a larger sample size with both hives. Next, we fed the bees to satiation. We did this by giving them access to a 50% mixture sugar concentration solution. And we fed all of them by hand, such as in this picture with a pipette up to their enclosure. And we did this with 1 full round and then a second full round to make sure that they were all at the same level of hunger or feeding before we started our experiment. Next, we incubated them for 24 hours. This step was either with or without treatment, depending on the experiment. For the first 2, with the fed sugar water versus unfed and the fed sugar water versus water, they were, in this case, incubated without their treatment. But with the last 2, the high versus low concentration and the poor versus rich nutrition, we gave them access to the food so that they would be able to feed during that time. But yes, they were separated in their two treatment groups at this point. And then from there, we transferred them into the drams after the 24 hours, either with or without their treatment had been applied. And from here, we organized the bees into their different groups. We made sure to keep them separate depending on their hive and their treatment group. And this was so that we could keep everything organized. And when we ended up applying the experiment, we did blindly organize the bee batches, if you will. And this was to prevent any bias from coming out as we made our observations. And then we now applied the heat stress. So this heat stress was done under 40 degrees Celsius conditions. It was applied for 5 hours and we performed 15-minute checks every 15 minutes. During these checks, we were looking for loss of motor control, what is called time to heat stupor. And this is essentially when the bee has, like I said, lost motor control and is not able to control its bodily functions, or it completely stays still altogether. And that was at the point where we decided that bee was no longer able to tolerate the heat. And then at the end of the 5 hours, we measured and recorded which bees had survived. So now looking at our results, first with the experiment fed versus unfed. Let me first explain the different axes. So we have time on the x-axis, which is representative in 15-minute increments. So 1 in this case would equal 15 minutes, 20 would mark the 5-hour point in this experiment, and so forth. With the y-axis, we're looking at the survival probability, and this is the survivorship of the bees in each group. With the yellow line on top, that is the fed group, and the blue line is the unfed group. And then the kind of clouds of shading behind each line are the 95% certainty markers. So with this graph, you can see that the fed group overall had a higher survivability, significantly higher with that. And the unfed group largely started to decline even from the start of the experiment, reaching their P50, which is their probability of 50% mortality or time to heat stupor, at about the 1 hour, 15-minute mark. And so you can see here that at the bottom where it is the number at risk, that's the number of survivors we had left at each of those checkpoints. And you can see it immediately drops off for the unfed group relatively fast compared to the fed group. Next, we looked at sugar water versus water, or the fed versus water group. And although the fed group doesn't have as clear results, because for the previous experiment, we had 100% survivability up until the 4-hour mark, which was remarkable. This does still show a significant higher survivability for the group that was given access to sugar water compared to the one that was only fed water. And with these results, we were able to eliminate the idea that the previous results were probably due to dehydration. Next, we looked at rich versus poor nutrition, again with the bee candy either containing pollen and sugar or just plain sugar. And these results we were actually surprised about. So we initially anticipated that the pollen rich nutrition source would allow for the bees to gain more energy and probably allow them to better tolerate the heat. But then when we conducted the experiment, we found that it was actually the sugar paste bees that performed better under these heat conditions. So this suggests that the sugar is what is important in their heat tolerance. And last but not least, we looked at high versus low sugar concentration, sugar water concentration. And as you can see from this graph, there is not a significant difference in the survivability between them. I also do want to acknowledge how there is a slightly more pronounced decline in their survivability despite their access to sugar water, but this was later in the summer, so it could possibly due to their just breeding, or if they had encountered heat sickness during this point in their hives, we weren't able to control for that. But regardless, with these results, we see that there's no significant difference, and so it doesn't necessarily matter the strength or the concentration of the sugar water that they're given access to. It's more so that they have access to which determines their survivability. And so what are the implications of this? So in this experiment, we used sugar as a substitute for nectar. And nectar is what they use as a source of energy and what they use, largely in their diets. For the implications broadly now, we want to go over the importance of the production of flowers as well as the bees themselves, largely in part because with environmental policies and actions, we typically tend to focus and zone in on saving one particular species, but through this experiment, by seeing the importance of nutrition and how it can greatly increase the survivability of bees experiencing heat, this will be important in terms of climate change with global warming. We will need to not only protect the bees themselves, but also the flowers that they depend on. In future studies, we might look at how we can control for age, because age was a factor that we weren't able to control in this experiment. As we were collecting bees from the hives, it was essentially which bees we could collect. And so next experiment, we might be able to, it might be better to control for that. Also, it would be important to look at different bee species, particularly because with our study, we only looked at Apis mellifera foragers. And so we should next suggest looking at different types of bees in terms of solitary versus primitively eusocial versus bees that are specialists versus generalists. We were looking at the Apis mellifera, which is a very generalist and a social bee. And they might have different thermoregulation habits than others. And then next, this is a little bit out there, but we would also like to look at the changes in flower behavior. And what I mean by that is the temporal changes in which flowers bloom, because with climate change, we might expect different blooming patterns in the flowers that are available and the times at which they choose to bloom. And so expecting or anticipating or studying how climate change might affect their blooming behavior might also help us prepare the bees for how they can better tolerate the global warming. And that is all for this presentation. We would like to thank the Malemi Hotel for hosting and supporting us during our research in Greece, also to the fellow researchers who made this study possible. I've listed our co-authors at the bottom right, and I have my contact information in the top left. Thank you, and if anyone has any questions, I'd be happy to answer them.